Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, so the last speaker of today and the conference is De Karp, who will talk about finding regulatory structure. And we're all very curious, in particular at Microsoft, what that might mean.
What we'd like to know is how to get rid of regulatory structure.
I see. Different kind of regulatory structure. Yeah. Well, first of all, I'd like to say that I've absolutely loved this conference, and I thank you very much for including me. I've learned something from every talk. It's been extremely stimulating. The area of phylogeny that many of the talks have concentrated on is one of the best formulated areas of computational biology and theoretical biology in a mathematical sense. And the topics that I'm going to discuss are have less of a pedigree. They are murkier. They're less well formulated. The algorithms are less refined. And so what I'm going to do is sort of speak in fairly broad terms, trying to introduce a number of topics that are of more recent vintage, telling you some results, showing you a few algorithms, but in many cases skipping the algorithms because you would be able to, there's nothing notable about them, and you'd be able to think up equally good ones on your own. And so in those cases, I'll concentrate more on the problem definitions and in some cases on the issue of how you would validate the algorithms. So this is probably unnecessary, but we can start by talking about the fundamental dogma of molecular biology, which says that in a eukaryotic cell, the DNA, which spells the names of the proteins, gets transcribed into the informationally equivalent form of messenger RNA, which gets transported out of the nucleus into the cytoplasm, where it gets translated into protein according to triplet code, which is universal and essentially universal in all living organisms. And it's the proteins that do all of the that regulate and perform so many of the key functions of the organism. And many of the topics, most of the topics in genomics have to do with understanding who the players are, which involves, for example, sequencing the DNA and identifying the genes, identifying the proteins that they create, understanding the mechanisms of regulation which allow the proteins to be produced in various quantities under various conditions, so understanding how transcription and translation and the subsequent activation of the proteins are regulated. And I want to give a quick survey of some of the goals and problems that have dominated genomics. First and foremost is the task of sequencing and comparing the genomes of many organisms, and now sequencing can be done extremely efficiently. And even though the genomes themselves don't do anything, they simply, so to speak, spell the names of the proteins, it's still essential to obtain them and then to process them to identify the genes and other functional regions within the DNA. Ultimately, we want to understand how all of these biomolecular molecules interact in networks and pathways and so forth to control the processes of the cell. That's going to be the focus of this talk. I don't need to say anything about tracing the evolutionary relationships of existing species. And then finally, a major area that's emerging into great prominence currently because of advances in sequencing technology and accessibility of data is the task of identifying the associations between genetic mutations and phenotypes such as disease. And this is really becoming a major industry with the ultimate goal of personalized healthcare. So this rapid development of this field has been fueled by advances in several fronts. The tremendous advances in genome sequencing. The first genomes that were sequenced were small bacterial genomes in the early 1990s, genomes of size of about 3 million nucleotides. Larger genomes were sequenced through the late 1990s. The key contribution of Gene Myers to whole genome shotgun sequencing changed the whole approach of the field. And now it's possible to sequence organisms very, very efficiently. So I'm probably exaggerating a bit, but it would be safe to say that that a current genome center can sequence a microorganism in a week, if not a day. Another front on which important advances have occurred is the area of measurement, large-scale measurement. The microarray technology permits one to intercept all of the mRNAs that are being produced in a cell or a collection of cells and measure their abundances, giving a key to how much transcription is taking place and allowing you to guess how much translation might be taking place. There are high throughput technologies for measuring other important interactions between biomolecules, protein-protein interactions, interactions between transcription factors and DNA through protein structures, protein abundances. Imaging techniques are advancing to allow one to observe the distribution of gene expression across a whole embryonic organism. Advances in databases make it possible for researchers to work with across the network with large data sets that they didn't generate themselves. And there are databases for genome annotation, which identify the genes, align several genomes against one another, databases of interactions between proteins and DNA, proteins and proteins, databases of metabolic pathways, and so forth. So there's a lot of data out there, and typically an investigation would look broadly at large amounts of data and data from different sources, which is a quite different approach to science than the earlier work at the birth of molecular biology, where a scientist would concentrate on data that was generated in his or her own laboratory and usually focused on a single protein or a very small system. Advances in mathematical modeling. I don't need to say much to this audience, but machine learning has been revolutionizing the computational approach to statistics, and combinatorial algorithms are quite important in this field. On this slide, I have a manifesto for the area that I want to discuss, which is basically going beyond the identification of the players, which is identification of the genes, identification of the proteins, and so forth, and going beyond the simple measurement of pairwise interactions between proteins or proteins and genes, and trying to find complex structures where many proteins and other biomolecules act in concert to regulate or perform some function. And I won't try to read this. You can read it for yourselves. But it's basically a manifesto which says that the future lies in discovering and characterizing the networks and the networks of networks that regulate and perform the functions of the cell. And so I want to talk about some specific problems within that general domain. So the first important point is that across the organism in different cells, different proteins are being produced in particular spatial and temporal patterns, and these are regulated by signals that are incident upon the cells and by the interaction of the proteins among themselves within each living cell. And so the expression of genes, which means basically the determination of how much of the gene gets transcribed and translated, is regulated by a complex network of interactions. And the key here is the word network, that we have to look at patterns of interaction among multiple entities rather than simply looking at the simplest pairwise interactions. So we have levels of regulation. We start with the genome that spells the names of the proteins. The first step is transcription of genes to messenger RNA. And a major investigation here is how the rate of transcription of particular genes, or the possibility of transcribing them at all, is mediated by proteins that bind near the start site of transcription of the gene. And according to the presence or absence of those proteins binding to the promoter region of the gene, it's determined whether and to what extent the gene is transcribed. Similarly, networks control the translation process and the subsequent activation of proteins to perform cellular functions. And finally, another level of regulation has to do with the actual chemical reactions that are catalyzed by enzymes to carry out the work of the cell. And then finally, if we're looking at diseases or other complex phenotypes, these usually involve participation from a number of different networks and have to be understood in the context of such interactions. So if you try to figure out why my nose looks just like my father's nose, it would involve quite a few different functional activities within the organism. So having given that embarrassingly simplistic introduction, which leaves out a lot of things, for example, such as the role of RNA in regulation and many new discoveries that are being made all the time, I now want to focus on a couple of more specific problem areas. The first of these, which will be the main topic of my talk, will be the attempt to look at databases of pairwise interactions between proteins to try to identify collections of proteins that work together to perform some functions. These will be of a couple of different types. They could be molecular machines or complexes which involve lots of interacting pairs of proteins, or signal transduction cascades, which are more linear structures where a signal enters a cell and then propagates through a chain of proteins or some closely linked chains of proteins, with each protein acting upon the next protein down the chain, ending up at one or more transcription factors that then control transcription. And so basically we want to look at these very noisy networks. Unfortunately, these networks are not terribly well validated or complete. They have lots of false positives and false negatives. But I want to talk about some of the problems that arise at looking at the data about which pairs of proteins bind together directly from a graph theory, thinking of that relation as a graph, and looking across several species, because we know that many of these pathways and networks are conserved across species, and trying to figure out what's going on, trying to home in by a kind of graph theoretic analysis on putative sets of proteins which can serve as putative molecular machines on the basis of the strength of the interactions and the similarity of the interactions across different species. And then more briefly, I'll talk about the analysis of protein-DNA interactions. This is the so-called cis-regulatory code, which refers to the surprisingly algorithmic processes by which the presence or absence of transcription factors binding near the start site of transcription determine the process of transcription. I'll say less about that second area, but I'll try to say a few words about it at the end of the talk. So just to recap what I've already said, the machinery that carries out and regulates the functioning of cells consists largely of sets of interacting proteins. There are other molecules involved, RNAs, small chemical molecules, but a lot can be understood by understanding how proteins interact in groups to form networks or pathways. And so these can be referred to as protein complexes or molecular machines or signaling pathways, which are more, as I described a moment ago, just more linear in terms of the dependencies among the interacting proteins. So we'll be dealing with two primary binary relations between proteins. One is interaction, by which I mean physical interaction, the actual binding of one protein to another. These interactions can be validated by experimental verification. There are techniques, so-called chip-chip techniques, or so-called two-hybrid experiment, or mass spectrometry. There are various methods of directly measuring and verifying that a pair of proteins bind to each other. There are also indirect inferences. So if proteins tend to be expressed under the same conditions, that gives a little more evidence that they may be physically interacting. If they're co-located in the cell, that can serve as confirming evidence. If they get inherited together, that similarly can serve. If they have many common interaction neighbors, that can also fortify the conclusion that the proteins interact. So somehow or other, people have been developing these databases of interacting proteins. A typical database for yeast, for example, might involve 6,000 proteins and some tens of thousands of interactions, but it would be very incomplete and very noisy. And that's one of the reasons why we have to go to comparative methods where we're looking at analogous interactions across different species can help to overcome some of the inaccuracy of the individual networks. And the second binary relation is a relation of similarity. What you would really like to know is which pairs of, and this has to do primarily with looking across species to try to find mechanisms that occur, say, in human and yeast or human and mouse, or even with remote mechanisms like the worm, based on the conservation of functional organization across, in the course of evolution, the conservation of functional organization across species. So what we would really like to know is in which cases two proteins in different species are orthologous, meaning that they derive from a common ancestral protein, and we can't really tell that very readily, so as a kind of substitute for that, we can use some form of similarity based on protein sequence or when available three-dimensional protein structure. So again, we have these noisy indications of which pairs of proteins can be thought of as, let's call them homologs, which might play the same role in two different organisms. So we can formulate this in graph theoretic terms. We can talk about the protein interaction graph of a species in which the vertices are proteins, and there are weighted edges which indicate the strength of evidence for an interaction between a pair of proteins. And these weighted edges are usually in the form of likelihood ratios, which indicate the some evidence of the conditional probability of the of interaction between the proteins given all the evidence. And so we can try to mine this data either in a single species or in multiple species. In a single species, we would work with the premise that highly dense subgraphs might be candidates to serve as molecular complexes. And of course, this wouldn't be conclusive. One of the things about this so-called systems biology area where you don't come in with a crisp hypothesis but simply scan very large masses of data looking for patterns of regularity, hoping that they have some biochemical meaning or biological meaning, will give you a lot of hypotheses. And a major issue is how you prioritize these, how you decide which ones should really be pursued and verified out of the many that occur. But the basic premise is that if you find that a collection of proteins have lots of credible interactions among them, then there's a better chance that they will have some common function, that they'll form some sort of complex. And similarly, if you can find a simple path, a sort of chain of distinct proteins where you have interactions pairwise between them, then it might be credible that the proteins in that path all lie in a single signal transduction network. So that's going to lead to some pretty complicated graph theoretic problems, let alone the basic scientific question of whether these premises are correct, that that if you get a dense subgraph, it's likely to have some reason for being dense, and so on. It gets even more complicated with multiple species, because there you have to consider not only looking for dense subgraphs or high weight paths in the individual species, but you would also like them to have ideally to be isomorphic under the similarity relation between proteins. So in the ideal case, you would find a complex in let's say human and a complex in yeast, and with the proteins being in a one-to-one correspondence under homology. And of course, that's not likely to occur, but you'd like to approximate that as much as possible. So let's now try to think about the kinds of mathematical and computational problems that we would have to face. And these are fairly canonical, well-known problems which are unfortunately hard to solve. So since we're looking for dense subgraphs, you might ask the following question. Given a protein interaction graph with weights on the edges, find a maximum weight connected subgraph with a given number of vertices. Well, that's kind of a variation on the clique problem, which is hard to solve and hard to approximate. NP hard to solve and NP hard to approximate. A little easier to deal with, and I'll talk about this in detail, is the problem of finding a maximum weight simple path with a given number of vertices. And this might arise if we're looking for plausible signal transduction pathways. Another problem is the hotspot problem. Can you find regions of a protein interaction network that contain many proteins of a particular type, proteins implicated and known to be implicated in some molecular function, or proteins associated with some disease, etc.
Why is it a given number of vertices?
Well, that's just a modeling technique. So you would let the number of vertices vary, but since there's only a linear number of vertices, you can chop it up into that restriction if you want to. What's it's a scalar number. What's the difference?
Well, whether you can sort of find by local changes.
Oh well, yeah. I mean, well, actually, what we're going to end up doing is use some heuristic which is not really worth describing in detail. It basically starts with some highly connected small seed and then keeps adding and deleting vertices according to some metric of density until you can't improve it anymore. But then beyond that, there's a question of validation. Is the score good enough to be able to claim that it couldn't have occurred by chance? We'll talk about that a little later. Okay, yeah, I mean, the maximal, finding a maximal one, a locally maximal one, is not NP-hard. Yeah. No, actually, on the contrary, if you allow repeated vertices, the problem becomes essentially trivial. It's because typically in signal transduction networks, you don't expect the same protein to occur twice along the.
But you have feedback and feed-forward loops in signaling.
Oh yeah, but what we're doing is taking a somewhat simplistic view in which we say that we want to think of this as as having a backbone which is really acyclic, and we want to think of the signal transduction network as being essentially a bunch of paths pasted together into a fairly narrow acyclic digraph. And yes, there would be some feedback loops, but we don't feature that in the analysis. So the hotspot problem is a little bit like some of these prize collecting Steiner tree and TSP problems that have occurred in the literature, and I was told yesterday that there's actually a factor 2 approximation for this problem. Although I'm not a big fan of approximation bounds which require a factor of 2 because we usually want to do better. But well, let me talk about one specific problem where one can do something nice. This is the problem of finding high-weight simple paths. So you're given a weighted undirected graph with a given number of vertices and edges, and you have a parameter t for convenience, where you're asking for a maximum weight simple path containing t vertices. So what would be the standard way of doing this? Well, the first thing that you would think of would be dynamic programming. So to formulate a dynamic programming algorithm, you ask yourself, if I'm generating some structure like a path, what is it that I have to remember about the path in order to figure out how the path could be continued into making a larger path? So that's, in dynamic programming, you're always sort of building up iteratively from smaller structures to larger structures. So what you'd have to remember about the partial path is which nodes are in it, because you don't want to repeat those in the simple path, and where it ended, because you have to hook it to the next node. So that suggests formulating a dynamic programming recursion based on a function of 2 arguments, a set of nodes S and a set of vertices S and a vertex within the set. And then you would want to tabulate for each such pair the maximum weight of a simple path ending in the given vertex and containing exactly the vertices in the set S. And then you can write down a dynamic programming recurrence, and you'll get an algorithm which unfortunately runs in time around n to the power t minus 1 times the number of edges in n to the t. So it's not too attractive. Yes? No, they're not. There's a whole literature on what kind of graphs they are, and they're scale-free graphs, roughly. They have some deviations from the scale-free model. They tend to have more triangles than you would expect in a scale-free graph. But the preferential attachment model is not too bad an approximation. So they're sparse graphs. But they tend to have some very high-degree nodes. So this, for t at all large, this algorithm isn't very attractive. But there's a cute randomized technique which allows us to find the optimal, the highest weight path of length, simple path of length t more efficiently. And this is based on a technique called color coding, which was developed by Alon, Euster, and Zwick in a a pure graph theory framework, but can be applied to this situation. So here's the cute idea that they came up with, and it was Noga alone who actually suggested applying it to us, that we apply it to biology. Randomly color the vertices. If you're looking for a path of length t, pick a uniformly random color from a palette of t different colors, and assign a random color to each node. And now, let's say that you, instead of asking for a simple path where the requirement is that the vertices be distinct, ask for a path in which the colors are distinct. If the colors are distinct, then the vertices in the paths will certainly be distinct, but on the other hand, there'll be certain simple paths where a color is repeated, and you won't find those. So you ask for a colorful path. You ask for the maximum weight path in which no two vertices are of the same color. And now when you write your dynamic programming recursion, all you have to remember about a set of vertices is the set of colors, which is from a t set rather than an n set. And so you get a much more efficient computation, but on the other hand, you might miss the optimal path because it might not be colorful under a particular random coloring. However, on the other hand, if you do enough random colorings, then you can be essentially certain that you'll find the best path eventually, that for one of them it'll be colorful. And moreover, along the way, you'll be getting a very broad selection of very good paths from the different colorings. So that sort of makes this a feasible calculation. You can write down the dynamic programming recursion, and the storage requirement is now linear in n. The execution time is linear in n and fairly moderate in t. You have to do it about e to the t times to make sure that the optimal path really becomes colorful in one of the trials. And you get the fringe benefit along the way of generating many high-scoring paths. So that we actually applied this to searching for putative signal transduction networks, and we were able to replicate some of the known signal transduction networks, sort of find them automatically by this technique. Another study that we carried out with the help of a protein interaction network was actually done by Ron Shamir from Tel Aviv and his student Igor Ulitsky, and then Ron came for his annual pilgrimage to Berkeley and discussed it with me, and we were able to simplify things a bit, so I ended up as a kind of courtesy co-author. This has to do with the following issue. People have been coming up with sets of proteins which can be thought of as the signatures for a disease, meaning that these proteins tend to be differentially expressed in individuals with the disease as compared with the general population. So this could be quite useful for diagnostic purposes. You could then use a microarray to measure the if you had one, a protein chip, to measure the abundance of these proteins, and it would give you a clue as to whether the person might be diagnosed with the disease. So for a number of diseases, these signatures have been propagated, and they've been found somewhat useful, but they're not terribly useful in finding the mechanism of the disease, because these proteins tend to be scattered around without interacting with one another. And so they don't form a pathway. They're just proteins that happen to be correlated with the disease. So in this work, Shamir and Ulitsky came up with the idea that you would like to find a disease signature which also had the property that the genes within it lay within a single connected subgraph of the protein interaction network. And these might then give more clue not only about the diagnosis of the disease, but the mechanism of the disease and possible drug treatments, drug targets for dealing with the disease. So, so we formulated the problem in the following way. Let's imagine that you have a number of individuals who have the disease, a typical one being individual X, and let A of X be the set of genes or the associated proteins that are differentially expressed in X, that are, that occur atypically in that particular individual. So then we can formulate the following problem. We would like to find a collection of proteins that have a high intersection with each of these sets A of X, but also lie in a small connected piece of the protein interaction network. So we formulate this question: given the human protein interaction graph, find a minimum cardinality connected set of vertices containing at least some target number of members of the set of differentially expressed proteins for each of the cases, for each of the diseased individuals. So this is kind of a set cover problem with the multiple set cover problem, because we want to cover each of these sets A of X at least t times, with the further requirement that you can find a sort of Steiner tree on the elements of your set cover that isn't too big. And of course it's NP hard, and we came up with some pretty good heuristics along the lines of the greedy multiple set cover algorithm, but respecting also the requirement that the novel requirement that the set of nodes that you choose, set of proteins that you choose, should form a tightly coupled part of the graph. So one of the tricks we used was the following: that if you're trying to cover each set a lot of times, like 15 times, then in the opening game of your construction of the set cover, you shouldn't be too fussy about which of these sets you're hitting. You should just sort of kind of score a node by how many of the sets it hits, and a protein by how many of the sets it hits. So in the first stage of the heuristic, we sort of score a particular protein by how many of these sets it hits, regardless of which ones they are. But then we have to have an end game where we use a more refined greedy algorithm to actually complete the covering of those sets that haven't been covered enough. And that worked pretty well. And of course, we had a little heuristic in there to find a small connected set of vertices that collectively hit a lot of these sets. That was done by a kind of breadth-first search. Yeah.
I mean, like often the Steiner tree problems, there are some worst-case guarantees. Do you have one here?
No. Well, yeah. I mean, a version of this heuristic inherits the logarithmic approximation ratio of the standard multiple set cover problem multiplied by the diameter of a local breadth-first search. So it's not very good, but it's no worse, really, than what you get in the ordinary set cover problem. So now I want to turn to perhaps a more exciting direction, where we're looking for interesting subgraphs, not just in a single species, but subgraphs conserved across species under this relation of similarity. So the rationale for this is that many complexes and pathways, most are conserved. They've evolved over evolutionary time, and they occur in modified forms in many organisms. And so you'd expect to see similar graph-theoretic substructures across species with orthologous proteins playing the same role in several of them. So so our goal is to identify such conserved structures, and one of the advantages of doing this is that we may then be able to transfer functional annotations across species. So if you know that two proteins interact in mouse, and then you could postulate that there's a good chance that their orthologs in human would also interact, and then you could go back and do a two-hybrid experiment to verify that. So it gives you candidates for discovering additional interactions. So how do we find, let's say in two species, how do we find interesting subgraphs that are replicated in the two interaction graphs? So this is a nice piece of work by my student Mani Narayanan, who's sitting out there somewhere. Right there, the guy with the smile on his face. He's a very brave man because he sprained his wrist falling off a bike, and he came anyway, and he's doing fine. What the idea behind this algorithm? It doesn't concentrate so much on the density of the subgraphs, but it's looking for a pair of subgraphs, one in the first species and one in the second species, that somehow locally look similar. There are a lot of problems of graph matching, finding matching subgraphs across two big graphs, which are NP-hard and hard to approximate. So it's amazing that Nani found a particular formulation which leads to a polynomial time algorithm. And the idea behind the formulation is that you don't insist on a bijection between the two subgraphs, but you simply require that in each of the two subgraphs, one from, say, human and one from yeast, every node in one of them looks like at least one node in the other, but you don't mind if it looks like several nodes in the other. So we define a notion given two subgraphs, H and K, connected subgraphs, connected induced subgraphs, under which we'll say that vertex U in H locally matches vertex V in K. And the requirement is that, first of all, U and V must be similar. They must be homologous proteins. And secondly, there should be some specified pattern around U on the one graph and V in the other graph, where the corresponding nodes of that pattern are also similar. And then you have different notions of local matching according to what that specified pattern is. So I give here the example of saying that U and V will be locally matching if there's a three-vertex path with U as the central vertex. Think of it then as a star with U as the center and two spokes, and a star with V as the center and two spokes, such that the corresponding vertices are similar. But you could allow other definitions. You could even relax the connectedness requirement, but I won't go into that. So then you could ask, so now we have a notion of two subgraphs matching in the sense that they're both connected and that every vertex in the one is locally matching at least one vertex in the other. And you could then ask for the maximal pairs in this relation. And it turns out that the number of them is polynomially bounded, and that there's a polynomial time algorithm for finding these maximal pairs. And the algorithm is actually extremely simple. It's a kind of a pruning algorithm. So you start with the complete interaction graphs. And you take the first one and you say, okay, let's throw away any nodes in the second one that don't match anything in the first one. Well, what may happen when you delete those nodes is that the second one splits into connected components. And then recursively, you take the first one against each of those connected components, and you go over to the component in the second one, and you say, okay, given that component, throw away everything in the first graph that doesn't locally match something in the component, and the first graph will split. And you go back and forth. And it sounds as if it could be exponential, but it isn't. It's polynomial because the graphs can't have too much in common as they split. And you get a polynomial time algorithm for finding these maximal matching pairs of subgraphs. And that's one way of looking for conserved structure across two graphs. we don't assume a unique one-to-one correspondence, but we assume that there's a many to many. So it's so it's just an arbitrary binary relation of similarity. The two sort of initial ventures into this problem of finding complexes conserved in a moderate number of species, 2 or 3 or maybe 4 at best, were NetBlast, discovered by my group at Berkeley jointly with Trey Eideker's group at San Diego, and the Tel Aviv group with Ron Shamir and Roded Sharan, and around the same time a project called Mawish, which was developed by a group at Purdue University. And I'm going to give you a definition which is a kind of mixture of the definitions that the two groups use. They're similar in spirit, but not in complete detail. So what you want, so you have these K species with their protein interaction graphs, and you want to find then K-induced subgraphs, which should be connected. You want to, and what you're looking for is something that you could call a conserved module, and that will depend on the densities of those individual subgraphs. You'd like them to have lots of edges, and on how well they correspond, how much similarity you can find under the homology relation among these subgraphs, and also whether the edges that lie in those subgraphs are in the corresponding places. Okay, so that's the spirit behind the definition, and then there are the details. So we can think, one way to formulate it is that you're looking for a set of connected induced subgraphs, and then you're also looking for a partition of the union of their vertex sets into blocks, such that within each block you have at most one protein from each species, and all of the proteins in a block are mutually similar. Okay, so you want groups of protein. You want these graphs to be dense. You want groups of proteins, one from each graph, to exist, which are all similar to one another or homologous to one another, and you want the edges to be in the same places as much as possible. So, from this you can come up with a scoring function where the score of a block is some increasing function of the number of mutually homologous species in that block, and the score of a pair of blocks is an increasing function of the weights and number of edges joining the two blocks, and the score of the subgraph is the sum of the scores of the blocks plus the sum of the scores of the pairs of blocks. So it's taking into account density, similar placement of edges, and similarity between proteins. And we want to find modules of maximum score, and we use a fairly prosaic local search to try to do this. And it works reasonably well. I'll show you a little bit of detail about the kinds of findings. The NetBlast group that I was included in worked with 3 species, yeast, worm, and fly, with 3 with their own protein interaction networks, and we used this technique to find fairly dense subgraphs conserved across these 3 species, and we found 173 putative complexes, and it turned out that the great majority of these had a uniformity of functional annotation. So if you go to these databases that provide functional annotations for the proteins, we found that in many cases there was a very striking homogeneity of functional annotation, which was encouraging and meant that we were finding something, possibly something very well known, but something. And here's a picture that sort of illustrates the kind of structures that we found, where the different colors represent the 3 species. You see a subgraph for each of the 3 species, and the dotted edges represent the homologies between them. So this is the kind of thing that we were searching for, and at least in the examples given here, there was a kind of uniformity of functional annotation among these, the nodes of these subgraphs. And one of the things we did, by the way, was to use these correspondences between species to home in on pairs of proteins that were homologs of interacting proteins in another species but had not themselves been found to interact. And then with the help of a laboratory in Stuttgart, we were able to verify that in about 50% of the cases, these unknown interactions, predicted interactions, actually occurred. And 50% is good because at random it would be less than 1%. Yeah. Well, the blocks would be indicated by, ideally by a triangle of dotted edges. But we also would allow blocks in some cases with 2 proteins. You'd get less of a score for having 2 than for having 3. I'm kind of running short of time, so let me just very quickly mention that a very nice piece of work came out of Stanford within the last year, another approach called Gremlin, which was similar in spirit, but which was aimed at finding conserved structure among many species, like 100 prokaryotic species. And that would be way beyond the methods that we developed with a view to working with 2 or 3 species. And the key points about what they did were, first of all, they found it convenient to assume that the homology relation was a transitive relation, in other words, an equivalence relation. That made it more convenient to carry out their computations. It's not, of course, literally true that it should be an equivalence relation, but they forced it to be an equivalence relation. So you can then think of each protein as having a color according to which equivalence class of proteins it lies in. They also assumed that they had a phylogenetic tree for the large set of species that they were working with. And their scoring function was more sophisticated than what we or the Purdue group used because it was based on this tree. So they had a similar situation where they had these subgraphs, one in each species that they were searching for. And they had these blocks, which in their cases consisted of vertices in the same equivalence class or having the same color. But then they would score the block according to the plausibility that those particular proteins would have been generated by the phylogenetic tree. So they were scoring according to mutations in the sequences of these proteins and also by duplications and extinctions of proteins. And so they got a more refined score based on this phylogenetic tree cutting across all these species, prokaryotic species. And they were also able to use the structure of the phylogenetic tree in their search for conserved structures because they would work up from the bottom of the tree, merging conserved structures as they went and carrying them as far up the tree as could be justified. So this was a nice advance. So I'm out of time. I'll just mention very quickly some things that I really didn't cover. One is sort of how you get a scoring function out of a probabilistic model. You would like to have more than an ad hoc scoring function. You'd like to have it model-based, and this can be done. Clearly, we need better heuristic algorithms for finding high-scoring modules. And then something I didn't touch on at all is after you've found something, how do you test its significance? And basically, we have used, and I think others have used, permutation tests where you sort of scramble the graphs while preserving some structure, like preserving the degrees of nodes. And then you run your algorithm not only on the true set of protein interaction graphs, but on a scrambled set of graphs. And you would expect that if there's biological significance to what you're finding, then you would get higher scores in the true graphs than in the scrambled set of graphs. And that's one of the statistical approaches that we used. But this means that you really need fast algorithms, because now you have to run not only on the real data, but perhaps 10,000 scrambled data sets in order to get any kind of p-value. And that means that the heuristics have to be very fast. So I never got to talk about cis-regulation, but time is up, so this is it. Yes?
In the case of single species, when you try to find these pathways,
Yes.
I mean, one of the issues is that each interaction itself might have very high confidence.
Yes.
However, the collection of all together might not have high confidence, because these might be coming from different experimental conditions, from different cell lines, from...
Yeah, that's true. That's true. This is something we didn't give very much attention to. We don't even know if the proteins are expressed at the same time. So most of the experiments are done in vitro, and it's very hard to know whether all of the conditions for these proteins to work together hold. So that's one of the many reasons why these are only putative structures. Yes, Joe?
This is almost the same question. In your graphs, your weights,
Yeah.
are they some sort of measure of likelihood ratio or probability of tail probability? Something, a statistical measure of some sort?
Yes, they are. They're, in fact, likelihood ratios comparing the likelihood of the data given an interaction with the likelihood of the data under a null model.
When you add up weights,
We're adding up log likelihoods, log likelihood ratios.
you're making an independence assumption, those are independent.
Yes, we have to make an independence assumption. We don't have to, but that's what we did. Yeah. 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 Okay, thanks. Yes, one more. Josie.
One general question. So you were mentioning at the beginning that most of the algorithms are heuristics.
Yes.
And so do you think that there is any value to theoretical, like worst-case algorithms at all in this area?
Well, I think approximation algorithms can be very interesting, not because of their ratios, but because you have to do something clever to prove to establish their properties, and that cleverness can probably be correlated with efficiency in practice as well as worst-case ratio. So the worst-case ratio of an algorithm by itself does not impress me, but if it's based on a very interesting algorithmic idea, it might be worth exploring further. That's my personal position on that. Okay, thanks so much.